Metabolism is a term that describes all the chemical reactions involved in maintaining the living state of the cells and the organism. These reactions are crucial as they allow organisms to grow, reproduce, maintain their structures, and respond to their environments. Metabolism can be broadly classified into two types: anabolic and catabolic reactions. Anabolic reactions are those that build larger molecules from smaller ones, whereas catabolic reactions break down large molecules into smaller ones, releasing energy in the process. Understanding metabolism is not just about understanding biology at a cellular level. It has profound implications for human health and disease. For instance, metabolic processes govern the energy we derive from food, which in turn affects our body weight, energy levels, and overall health. Disruptions in metabolic processes can lead to diseases such as diabetes and obesity. Thus, a clear grasp of metabolic pathways is crucial for anyone looking to understand human health deeply. Metabolism is a dynamic and continuous process. The human body adjusts its metabolic processes based on various factors such as nutrient availability, physical activity, and energy requirements. This adaptive mechanism is vital for survival, helping to maintain a balance in energy production and expenditure. By converting nutrients from our diet into energy, metabolism fuels all bodily functions, from the simplest cellular processes to complex brain functions. Apart from energy production, metabolism also involves the removal of waste products that accumulate in cells, thus preventing potential harm. These waste products are the result of various metabolic reactions and, if not properly managed, can lead to cellular damage or disease. Therefore, understanding metabolism is not only about knowing how energy is produced, but also about how it is used efficiently and safely by the body. Anabolic pathways in metabolism are concerned with building complex molecules from simpler ones. These reactions are crucial for growth and repair processes in the body. For example, during muscle repair or growth, anabolic reactions help synthesize protein molecules from amino acids. Another instance is the synthesis of glucose from non-carbohydrate sources, a process known as gluconeogenesis. These pathways require energy, generally derived from ATP, the energy currency of the cell. On the other hand, catabolic pathways involve the breakdown of complex molecules into simpler ones, releasing energy that is stored in ATP. A quintessential example of a catabolic pathway is glycolysis, where glucose is broken down into pyruvate, generating ATP and NADH, crucial energy carriers. Another important catabolic process is the breakdown of fatty acids, which serves as a significant energy source during prolonged fasting or exercise. The balance between anabolic and catabolic processes is tightly regulated and essential for maintaining homeostasis in the body. Hormones such as insulin and glucagon play a significant role in this regulation. Insulin promotes anabolic processes, helping in nutrient storage and growth, while glucagon stimulates catabolic processes to release stored energy in response to low blood glucose levels. Bioenergetics, a subfield of biochemistry, studies the transformation of energy in living organisms, connecting it to anabolic and catabolic reactions. By understanding how organisms convert food into energy, bioenergetics provides critical insights into how genetic or environmental factors could impact health. For example, a better understanding of bioenergetics can help in designing diets that optimize energy use and storage, crucial for managing conditions like obesity and type 2 diabetes. Carbohydrate metabolism involves several key pathways, the primary of which is glycolysis, followed by the TCA cycle and oxidative phosphorylation. These pathways are central to energy production in cells. Carbohydrates, primarily in the form of glucose, are the body's preferred source of fuel. After ingestion, carbohydrates are broken down into glucose, which is then available for use as energy, stored in the liver and muscles as glycogen, or converted to fat. The initial step in carbohydrate metabolism is the uptake of glucose into the cells. This process is facilitated by insulin, which signals cells to increase glucose uptake and utilization. In the absence of insulin, as in type 1 diabetes, glucose cannot enter most cells, leading to elevated blood sugar levels and metabolic complications. This highlights the importance of insulin in maintaining normal blood glucose levels and energy metabolism. Following glucose uptake, glycolysis begins in the cell's cytoplasm, where glucose is converted into pyruvate. This process produces small amounts of energy captured in the form of ATP and NADH. These energy molecules are then used in further metabolic processes to produce a significantly larger amount of energy, essential for all cellular processes. Glycolysis is followed by the TCA cycle and oxidative phosphorylation, where the real energy production occurs. These stages are highly efficient and responsible for the majority of the ATP produced in our cells. Hence, understanding these processes is crucial for anyone studying biochemistry, health sciences, or medicine. Glycolysis is a fascinating and vital metabolic pathway that represents the first stage in the breakdown of glucose to extract energy. It occurs in the cytoplasm of cells and can proceed with or without oxygen, making it an anaerobic process. Glycolysis converts glucose into two molecules of pyruvate, producing a net gain of 2 ATP molecules and 2 NADH molecules, which are used in later stages of cellular respiration to generate a significant amount of additional energy. The process of glycolysis involves 10 enzyme-catalyzed steps, each carefully controlled to maximize energy extraction. The first 5 steps are known as the energy investment phase, where 2 ATP molecules are used to modify glucose, making it more reactive. The following 5 steps, known as the energy payoff phase, generate 4 ATP and 2 NADH molecules. This careful balance ensures that energy is both invested and harvested efficiently during glucose breakdown. One of the critical enzymes in glycolysis is phosphofructokinase, which catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. This step is a key control point and is heavily regulated by ATP levels, thus directly linking glycolysis to the energy needs of the cell. When ATP levels are high, the enzyme's activity decreases, slowing down glycolysis. When ATP levels are low, the activity increases, speeding up glycolysis. The end product of glycolysis, pyruvate, has several fates depending on cell type and oxygen availability. In the presence of oxygen, pyruvate enters the mitochondria to be processed in the TCA cycle. In anaerobic conditions, it is converted into lactate in a process called lactic acid fermentation, which is crucial for regenerating NAD+, allowing glycolysis to continue producing energy even without oxygen. This flexibility makes glycolysis a crucial metabolic pathway in energy production under various physiological conditions. Section 5. The TCA cycle, completing glucose oxidation. The TCA cycle, also known as the Krebs cycle or citric acid cycle, is a fundamental component of cellular respiration, located in the mitochondria. It represents a crucial stage in the metabolic breakdown of carbohydrates, fats, and proteins. Here, the products of glycolysis, pyruvate, are completely oxidized to produce carbon dioxide, ATP, and high-energy electron carriers, NADH, and FAD2, which are crucial for the next stage of energy production, oxidative phosphorylation. Upon entering the mitochondria, pyruvate undergoes a transformation into acetyl-CoA, a process catalyzed by the enzyme complex pyruvate dehydrogenase. Acetyl-CoA then enters the TCA cycle, which involves a series of reactions that not only generate energy but also produce key molecules that serve as starting points for other metabolic pathways, including the synthesis of amino acids and fatty acids. Each turn of the TCA cycle yields 1 ATP, or GTP depending on the cell type, 3 NADH, and 1 FAD2. These electron carriers then transfer their high-energy electrons to the electron transport chain, driving the production of a significant amount of ATP through oxidative phosphorylation. This efficient energy production system is critical for high-energy demanding processes, such as muscle contraction, active transport across cell membranes, and biosynthesis. Moreover, the TCA cycle plays a pivotal role in the metabolic integration of various nutrients. It is not only the final common pathway for the oxidation of carbohydrate, lipid, and protein molecules, but also provides intermediates for various anabolic processes. This highlights the central role of the TCA cycle in both energy production and the synthesis of crucial biomolecules, making it a key area of study for understanding cellular metabolism and its implications on overall health. Section 6. Lipid metabolism, fatty acids, and energy. Lipid metabolism is another vital aspect of the body's biochemistry, dealing primarily with fats and fatty substances. Lipids are crucial not only as a major form of energy storage, but also in forming cell membranes and acting as signaling molecules. The metabolism of lipids involves two primary processes, lipolysis and beta oxidation, which break down triglycerides into fatty acids and glycerol and then further oxidize the fatty acids to generate energy. Lipolysis, the first step, occurs in the adipose tissue, where enzymes like hormone-sensitive lipase catalyze the breakdown of stored triglycerides into free fatty acids and glycerol. These free fatty acids are then transported to various tissues, including the muscles and liver, where they undergo beta oxidation. Glycerol can be converted into glucose via gluconeogenesis, providing energy when carbohydrate intake is low. Beta oxidation of fatty acids takes place in the mitochondria of cells. Here, fatty acids are broken down into acetyl-CoA units in a series of reactions that also produce NADH and FADH2. These molecules are then used in the TCA cycle and electron transport chain to generate a substantial amount of ATP. This process is particularly significant during extended periods of fasting or low-carbohydrate diets, where fats provide the majority of the body's energy. Moreover, the regulation of lipid metabolism is intricately linked to carbohydrate metabolism. Insulin, for instance, plays a significant role in inhibiting lipolysis and promoting fat storage, particularly after meals when glucose levels are high. Conversely, during fasting, glucagon levels increase, promoting lipolysis and beta oxidation to maintain energy supply. Understanding these regulatory mechanisms is crucial for managing diseases such as obesity and type 2 diabetes, where lipid metabolism is significantly altered. Section 7. Beta oxidation of fatty acids. Beta oxidation is a metabolic process involving the breakdown of fatty acids to generate acetyl-CoA, NADH, and FADH2. These products are essential for the TCA cycle and oxidative phosphorylation, which are part of cellular respiration. The process of beta oxidation takes place in the mitochondria and starts with the activation of fatty acids in the cytoplasm, where they are converted into fatty acyl-CoA by the enzyme acyl-CoA synthetase. Once activated, the fatty acyl-CoA is transported into the mitochondria via the carnitine shuttle. Here, the fatty acid undergoes a series of dehydrogenation, hydration, and thiolytic cleavage reactions to break down into acetyl-CoA. Each cycle of beta oxidation shortens the fatty acid chain by 2 carbons, producing one fatty H2 and one NADH in the process, which are then used to generate ATP in the electron transport chain. The efficiency of beta oxidation is evident in the high ATP yield per molecule of fatty acid. For example, the complete oxidation of one molecule of palmitate, a common 16-carbon fatty acid, yields 129 ATP molecules. This high-energy output makes fatty acids an excellent energy reserve, especially during prolonged physical activity or periods of fasting. Moreover, dysregulation of beta oxidation can lead to metabolic disorders. For example, an inability to adequately break down fatty acids can result in the accumulation of fatty acids or their derivatives, which can be toxic. Disorders such as fatty acid oxidation disorders, FAODs, highlight the importance of understanding and regulating this pathway in maintaining metabolic health and preventing disease. Section 8. Amino acid metabolism. Amino acid metabolism is a critical part of overall metabolism, involving the breakdown and synthesis of amino acids, the building blocks of proteins. This metabolic pathway is not only essential for protein synthesis, but also plays a significant role in producing energy, especially during times of starvation or intense exercise, when protein is used as an energy source. The breakdown of amino acids begins with deamination, the removal of an amino group, resulting in the formation of ammonia and a corresponding keto acid. Ammonia is converted into urea in the liver and then excreted from the body, primarily through urine. The remaining keto acids can enter various metabolic pathways, including the TCA cycle, to produce energy or serve as precursors for the synthesis of other compounds. Amino acids are also pivotal in synthesizing hormones and neurotransmitters, illustrating their importance beyond mere components of proteins. For instance, the amino acid tryptophan is a precursor for the neurotransmitter serotonin, known for its role in regulating mood, appetite, and sleep. Similarly, tyrosine is involved in the production of dopamine, another vital neurotransmitter. Moreover, amino acid metabolism interacts closely with other metabolic pathways. For instance, several amino acids can be converted into glucose through gluconeogenesis during prolonged fasting, providing a vital energy source. This interplay highlights the integrated nature of metabolism, where pathways are not isolated but interconnected, impacting overall health and biological functions. Section 9. Metabolism and Health: Why it all matters. Understanding metabolism is crucial for more than just academic purposes. It has a profound impact on our health. Metabolism influences everything from how we use food to how we maintain weight and how our bodies respond to disease. For instance, metabolic disorders such as diabetes and obesity are directly linked to disruptions in normal metabolic processes. The study of metabolism also provides insights into how we can manage and potentially cure metabolic diseases. For example, dietary interventions that manipulate metabolic pathways can significantly affect body weight and energy levels. Similarly, understanding the metabolic pathways influenced by different drugs can help in the design of more effective treatments for diseases like type 2 diabetes. Furthermore, the emerging field of metabolomics, which involves the comprehensive analysis of metabolites in a biological system, is providing new insights into metabolic changes associated with diseases. This can lead to better diagnostic tools and more personalized medicine approaches, potentially transforming how we treat complex diseases. Finally, promoting a broader understanding of metabolism can empower individuals to make informed decisions about diet and lifestyle that support optimal metabolic function. By understanding the basic principles of how our bodies process and use food, we can make choices that enhance our health, well-being, and longevity. The intricate world of metabolism, therefore, is not just a topic for scientists and clinicians, but is increasingly relevant to everyone interested in maintaining their health and preventing disease.